All right, well, first of all, I want to thank the organizers for another opportunity to speak at a biopharma event. I do remember there was one a little earlier in the West Coast, more on the clinical applications, but it was just as well attended, so thank you again. So yes, I'm going to try to spend 15 minutes, leave enough time for questions, to talk about GMP, which is almost impossible to do without putting people to sleep. So I'm going to try not to bore everyone with this. I am from a contract research organization, and I'm going to be talking about good manufacturing practices with the little c, current good manufacturing practices. It not only applies to manufacturing, but it also applies to those of us who are in the service industry. We have to find very creative ways to make things GMP when we're working with vendors who like to have research use only or RUO instruments in the world. So I just wanted to give a little bit of a crash course here of the merging of a few different areas or of a historical timeline and how it's really important in this space, including the tools that we use. So this is one of my favorite slides. If you think about it, this is a long history of things going on in the world with not only sequencing, the emergence of DNA, understanding of DNA, overlaying that, pharmaceutical production, biosafety testing, and so forth, and just trying to keep pace with things. So at the very top of this is actually a timeline from 1901 to 2018 with some critical events in the world in which manufacturing or the application of therapeutics have had actually a rather negative impact on some patients. And in fact, it has led to multiple deaths. And this includes the history of Jim the horse and the serum being contaminated with tetanus, the diphtheria toxin, all the way through to even current events. So there's a 2018 study in there with about a vaccine in China in which there were some deleterious effects there. But if you look at this, so it's really, it's a fascinating timeline to look at, and if you try to overlay where the health authorities add to this, if you're looking at, and I have my cheat sheet here for dates, so, and I want to say thanks to some of the earlier speakers who kind of positioned this. In 1848, in the US, they started with the safety of agricultural products. That turned out into 1906 to the Pure Food and Drugs Act to try to clamp down. As you can see, we had some early events to try to clamp down on the safety of products. In the 1930s, 1930 itself, there was actually a name change for this, and it used to be called the Food, Drug, and Insecticide Administration. They decided very smartly to get rid of the word insecticide and move to the Food and Drug Administration. In 1938, so again, through part of this timeline, that's when there was another significant change, and that was the Food, Drug, and Cosmetic Act. In the 1940s is when GMPs really came into being. So in 1941, they only cared about purity and potency. Only in 1945 did safety and efficacy come into play. So before that, it was just product. After that, oh, maybe we should think about the patient, because if we don't, we won't be able to sell products to anyone because we won't have any patients. Then in the early 1970s, late 1960s is when Europe really got on the bandwagon and started to compile all of their interest in GMP into some documents, and even as late as 1978. And a lot of the stuff that we work with today is actually still based on documentation from the 90s. And this might predate a lot of us, but it's just a fascinating foray to see when things start to change. In my industry, sequencing became really pivotal 20 years ago, almost 25 years ago now. So the era of sequencing, next-gen, long-read, nanopore sequencing, you name your sequencing, really started in the early 2000s, and it continues to today. And you can see there's been a big challenge to keep the price of the genomes down, to keep advancing testing technologies in light of some of the newer modalities that are out there. And you heard a great case about the COVID-19 vaccine. How do you have a rapid development of a vaccine and keep it GMP compliant and have rapid testing as well? So all that really ties together very nicely in this slide here. And one of the curious things that I always like to note is it took 4 years for the price of the genome to come down $99. So, but moving on. So in my space, so I work in the biosafety testing space. And I'm an R&D-er working in this space, and I started in this space back in 2008, and I knew nothing about GMP at the time, but I had to learn very quickly because I'm being told, you need to know this, you can't just do what you want. And I thought, well, that's no fun anymore, but I get the idea. So really dabbling in this next level of sequencing at the time. And if you look on the left-hand side of the slide, you can see there's different elements in which we have to test against our therapeutics against, and these are dictated in regulatory guidance. So the global health authorities and regulatory bodies have come up with all kinds of documents, then there's a whole slew of them, with recommendations for testing for your products to ensure the safety. So they just said, well, we want you to do this, but we're going to hold your feet to the fire, and we're not going to tell you how to do it, but we just really want you to do this. And if you say, nah, no, they'll say, no, we're going to make you do this, and then you have to come up with ways to creatively do that. So we know that these testing recommendations are found in these global regulatory guidance documents. We have to follow them, but we're making our own way as to how we're going to follow them. Now, if you look at some of the classical tests that are out there, for those who are less familiar, you have animal-based tests, so these in vivo tests. And there is a current push to get away from animal-based tests, so the 3 R's principles. We have these cell-based tests, or these in vitro tests, again, looking for adventitious agents. Mostly it's virus hunting. You want to make sure there's, and you'll hear more about this with Serge today, you just want to make sure that there's nothing else in your sample that could make a patient sick. And then, of course, there's a lot of molecular-based assays that have their place in there. But if you're looking for someone like me and you think, well, I do sequencing, I have a sample, I have a sequencer, I have data, I can answer most of these, right? And the reality is, yes, we probably can, and we can probably do it, a lot of it into a single test as well. But it has to be a GMP-compliant test. So where are we today? Okay, acronyms. You get buried in acronyms. So sequencing in various modalities is explicitly included in these international regulatory filings. And the most recent one to be updated was the ICHQ-5A, which is the International Consortium on Harmonization, blah, blah, blah. And they have a series of these documents. They just updated it. They have included sequencing now as a very specific testing strategy here. So we have a lot of clients now going, now we want to use sequencing, and it has to be GMP, and it needs to support my IND or BLA, so your investigational new drug, biological license agreement. But I need it to be GMP, I need it ASAP, and while you're at it, do you have a biological master file that I can reference, or a drug master file that I can reference? Because I don't want to look through all your data, but I want to see your validation packages, and those things are just massive documents. So that's what we keep getting asked for. So where do we start, right? First of all, you have to understand what kind of test you want to do. Is it a qualitative test, limit test, quantitative test? These all matter because all of the features, the testing attributes, accuracy, linearity, range, precision, robustness, specificity, limit of detection, you get to, depending on whether you're qualitative, quantitative, or somewhere in between, will determine how many of those you actually have to meet from a testing strategy, how many features of that, and that can drive how big a validation package can get. Then, of course, you have to understand what regulatory requirements you need to follow. The ICHQ-2 is probably one of the master documents. There is a new, there's another new chapter. The EFPIA has just put out how to validate sequencing assays. It's in draft right now, or it may have just recently been issued. You have the ICHQ-14, another iteration of ICHQ-2. We have all the European documents. We have your Annex 15s from Europe. You have your ISO requirements. You have your WHO requirements. It just goes crazy like this. And not everyone is aligned with one another. Some say you can test this way. Others say, no, not really, not here. So you have to try to align all of these things. But right now, most in the industry are following the ICHQ-5A requirements. And that's great, because that governs your test. But you're not just thinking about a test. You also have to think about validation of equipment, which is not a trivial task when it's all research use only or considered portable. MinIONs are portable instruments. Makes it easy to validate, but then you have to think, well, it's portable. If I lose it, I get another one, but then I have to reinstall, re-IOQ it, and so forth. So there's some considerations to be taken there. And then, of course, the biggest consideration in the field of sequencing is, of course, you have massive amounts of data. What are you going to do with it? How do you maintain integrity, security? How do you move it to the cloud or not? Can you use cloud-based resources? You can in the US, no problem. In Europe, they're a little bit more fidgety about these things. They don't like that very much. There's all your GAMP requirements. again, there's just a number of requirements that you have to think about that go into all of this. So computerized system validation, I think, has been an interesting foray, I think, for the nanopore community as well. Nice to see the Q-line instrument, but someone like me struggles with the Q-line instrument simply because I still want to do R&D, but now you've locked me down in an instrument for GMP. And they don't always coexist. So it's a bit of an internal struggle I got to deal with, but it's always been in that world. Sequencing has always been a world like that, where you have to mitigate changing chemistries, evolving equipment. Oh, there's a software upgrade, just do it. No, I can't. I have to do change control, and I have to then document what the revision history is, and I need to know, and I have to test against the new features. It's not just, oh, I just press the button. And in full disclosure, we did have somebody in our lab who thought, oh, look, there's a software upgrade, and went, ding. And then I thought, oh, okay. His name is Crispin, if you want to. Crispin? Crispin, we weren't supposed to do that. I understand your R&D, but that was a no-no. Oh, what do we need to do now? We need to go down a whole foray of how to get it back to where the instrument was when we validated it. So again, not necessarily trivial, but it is fun to do when you have a lot of changing variables that change very, very quickly. Remember, this is a science that has to evolve very quickly. You have therapeutics. You saw from the earlier talks, you have all of these great targets to aim for in terms of vaccines, therapies, and so forth. You need to have the science evolve, but how do you do it in GMP? Because remember, when you do GMP, it's not just locked down because I want to lock it down. Our clients go in and say, I now have a filing with the FDA that says this is my test method, and you cannot change that. And I said, well, I have an update. I have better chemistry. It's going to be so cool. It's much better. No, it's going to take us 6 months after you give us notification to even think about changing it. So you have to maintain it in legacy. So we have to have, and our requests, of course, are, can you maintain a kit, a reagent, a flow cell in legacy? And we get the, well, we don't want to. Like, because that's revalidation, and that's a whole other foray into validation. Okay, so I'm going to talk specifically about an assay. You've already seen elements of this morning. And this is our integration site analysis. So there is a guidance document out there, which is the ICHQ5B. Unfortunately, I know these numbers and letters now very well. It's a guidance document that says that if you're manufacturing, particularly a monoclonal antibody, that you use a Chinese hamster ovary cell or system to develop it in, you want to characterize where it is in the genome so you know where it is, how many copies are there, what the configuration of the copies are, and all of these other elements that you can use a whole series of classical testing to do. And there's like 7 tests to do, not always ideal. Whereas you can do it in a single nanopore-based sequencing test, and the key is, of course, you need long reads to do this. So effectively, it's a 2-round test where you use CRISPR-Cas mediated mechanism, which you've already heard this morning. You target the region of interest, and you sequence out, and you figure out where it is in the genome. And every other genome other than a hamster, that's easy to do. Someone threw a hamster in the mix. Hamsters, all right, so CHO cells are great for production. They're great. You can get high titers. They're really well characterized. They've been used for decades, longer than I am old. They're great. They're horrible to characterize. Right, because their genomes, you look at the hamster sideways, and its genome goes, and it starts rearranging. And now it's not chromosome 1 anymore. It's chromosome 1 with a bit of chromosome 4 and some 7 thrown in backwards, and then they deleted this one. And if you look at, you have to mine data and compare it to reference sequences. It's really hard to do when the thing keeps moving around. But you're trying to build a GMP assay that fits not just one client in the industry, but every client in the industry. So that in itself is another challenge. So we do this, it's inside-out sequencing, and then, of course, we can flip it around once we have, and apologies for the obstructed text, but you can flip it around and do outside-in sequencing to then characterize the insertion site. The actual vector itself. And those vectors do all kinds of crazy things because people use plasmids that are grown in E. coli and they don't purify them and they get incorporated. The E. coli gets incorporated into your genome while you're at it, and it just makes a really simple insertion, hundreds of kb long instead, with a lot of different elements. So anyways, we do use we use a GridION system specifically for this, and it allows us to very specifically look for where these vectors are in the genome. This is great. It's a required test in the industry, so it must be GMP. Okay, so adventures of GMP validation. We now have this, that's our test. We want to validate that test. Okay, we built a test on R9s, flow cells, and earlier chemistry. Great, we now need that to be stable. We have our test methodology. It could be any cell type. We have to have GMP certifiable controls. We have to make sure our reagents are available for eternity for this test. We have to do vendor supplier qualification. We have to understand what our assay limitations are, what we can and can't do. We establish our acceptance criteria, our system suitability testing requirements for different aspects of the test and so forth. So we have all of that figured out. We now need to validate that part of the test. We have our equipment. Don't underestimate the need for audit trails with the FDAs these days. Okay, so 2019, they really were a stickler for data integrity, and they created some new documentation that requires that. And if you're in the, again, if you're in the sequencing field, data is our product. Or same thing, unique user logins. You can't just have a generic one. Have to maintain Part 11 compliance, and you have to be able to move data around pretty quickly. And storage, where do we store everything? And then, of course, for the analysis methodology, what GAMP category are we in for everything, Part 11 compliance? Software as a service, very interesting situation. How do you validate somebody else's software when it's downloadable or you're using their web-based application? That can be a challenge in itself. Or is it your custom service? Again, I already mentioned cloud-based infrastructure. It's great to use, but I have had in my past the incident, and I'm running out of time now, where use actually Google as a source, and they had a failure. And although they said we lost 0.00004% of our data, they still lost data too. Remember, they're just server farms sitting out someplace out west. And then separate validations for schemes. We tried doing this as a concurrent validation at 2 different sites. So we required 4 sequencers, 4 operators, all of our ancillary equipment to be validated, harmonize logistics on this, harmonize materials shipped across the world in order to make that happen, but very successful. So here, again, just document overload. I won't go through this. This is actually a test strategy. So 11 test scenarios there done at both sites. Not trivial, but the finish line is to achieve a GMP compliant test here. So I want to go through this because this is the fun part. Things you don't think about that always happen, and I've done this enough times to see it. First of all, be careful of your certified reference material. We've had instances where it wasn't what it was supposed to be from a vendor. This is certified reference material. They did not properly certify it. It was something completely different. My other one that I'd love to happen is when the network just kind of craps out in the middle of data transfer, and you go like, I don't know how much I lost. I don't know where it went. Yes, as I mentioned, audit trails, individual user logins are required. Everyone says, did you validate this sample type? There's no way I can validate every single sample type. I want to do representative validation as I can. This one, for this specific crowd, yes, we do need expiration dates printed on packaging. That is a critical feature. Otherwise, we make them up, and that becomes a mess. I just bought and IOQ'd an instrument, and it's already obsolete. We've had that happen. We've had instruments become obsolete before we even get them out of the box and had to return them to the vendor for a new model. This one has happened. Windows 10 machines update by themselves to Windows 11, even though you try to put protections in place, and it bricks it. Not only does it brick it, when you try to unbrick it, it can cost a considerable amount of money because you have to do IOQs on that. And then, of course, the famous one here is, why do I need to buy a permit? Why do I need a permit to buy ethanol in Pennsylvania? Apparently, there's a lot of drunks in Pennsylvania that they need to worry about. But these are elements that you have to keep into consideration here. So while it touched a bit on GMP, I have less than a minute. That is my last slide here. I won't go through this, but it can definitely be done. Working with the agencies, it can easily be done. This is a platform that can easily be validated for GMP compliance. It just does require a bit of consideration.